Hi everyone, I am Dr. Anisha, your microbiology faculty, and today I will be taking up a very important topic for your exams point of view, that is Vibrio. Now before getting into the details about the pathogenesis and the other complications, we first need to understand few basics about this particular bacteria. Now Vibrio, firstly it belongs to family Vibrionaceae. And Robert Koch in around 1886, he named this bacteria as 𝗘𝗵𝗄𝗃 because of its curved nature, and comma-shaped appearance. Comma-shaped appearance, right? So we describe its morphology as gram-negative, rigid, curved bacilli, which are motile with the help of a single polar flagella. Right? So can you appreciate this in this particular image? This curved nature of the bacilli as well as a flagella coming from its pole. So it's a single flagella and it is helping the bacteria in its motility, and one flagella coming out from one side or from one pole, right? So a very important question for your exams, it is very frequently asked, the type of motility that is depicted by this particular bacteria, and it is one of the favorite questions from this topic. The type of motility is darting or shooting star motility. Darting or shooting star motility, right? So most of you might have at least once in your lifetime experienced this shooting star-like situation. See how fast in this GIF the star is moving. So how rapid the movement is. So, and it is so rapid that you blink and you miss. And so does we call it as darting motility. See how fast we throw these dart pins onto the board, right? And that is exactly the case of this particular bacteria, Vibrio. It moves so rapidly that there's going to be a significant change in position of the, there's going to be a significant change in its position when we observe it under the microscope, right? And it is very clearly visible under microscope. So there is significant change in position, right? So in order to observe the motility of this bacteria, we do a special preparation, that is hanging drop preparation. And how do we do it? We either are going to take a concavity slide or we are going to take a normal slide and we will make a ring out of the molding clay. And now we take, this is a slide where we have this paraffin ring or molding clay ring, and onto a cover slip we will put up a drop of the stool sample and we are going to invert the slide onto the cover slip, and then we flip the cover slip. So this is what we get, the kind of interface. See, we get the drop which hangs from the cover slip and we observe it under the microscope. And what place are we going to observe the motility of the bacteria? It is going to be at the edges of the drop. So why do we consider looking for motility at the edges? Firstly, because it provides a better contrast. Secondly, majority of the motile bacteria, they tend to move towards the edges of the, of the drop. See, a lot of bacteria, these are seen coming towards the edges of the drop. And why is that happening? Because of higher oxygen tension at the end of the drop. So they tend to move towards higher oxygen tension And this rapid motility, this darting or shooting star motility, it is very easily visible onto the drop. So one bacteria, they move so quickly that they're going to change their position like this. They're going to change very rapidly, right? So this can be very easily observed under the mic, right? So now we know that Vibrio, it is naturally a salt-loving bacteria. So the habitat are also going to be the places or the bodies that contain higher salt concentrations. So firstly, the habitat is going to be seawater, of course, seawater, rivers, seafood would be rich in salt, right? And all the bodies which contain higher salt concentration, so that will be naturally the habitat of Vibrio, right? So now Vibrio cholerae is one such bacteria that has been notorious with a lot of pandemics and epidemics worldwide, leading to a special type of stool, that is rice water stools. So it is very important to make an early diagnosis of this disease and for good epidemiological purposes as well. So we need prompt diagnosis and prompt treatment because this bacteria has been responsible for n number of epidemics throughout the world, and of course pandemics, right? So, and it also is responsible for the rice water stool diarrhea. It is an acute condition, so it is responsible for acute diarrhea, acute diarrheal disease, right? So now moving towards the classification of Vibrio. So firstly, these can be classified, Vibrios can be classified firstly on the basis of salt requirement, on the basis of salt requirement, right? So these Vibrios can be classified as halophilic, as well as non-halophilic. Now, halo or loon. Now, loon is a word which is used to describe salt in certain Kumami regions of Uttarakhand, if you might be knowing. So yes, these halophilic Vibrios, they require loon or salt. Loon, that is a mnemonic that I've kept for you guys to remember the halophilic Vibrios, that these guys, they require salt for their growth. So salt is an utmost necessity for these Vibrios. So, and the concentration of salt that is required, minimum concentration that is required for the growth of these bacteria is about 7 to 10% salt concentration is optimum. They can tolerate as well as grow in this salt concentration, right? They can tolerate as well as grow at this salt concentration. And these non-halophilic Vibrios, they do not require salt for their growth, right? No salt requirement. Do not require salt, but a particular minimum concentration of salt is optimum, again optimum for their growth, right? And that salt concentration is around 1% of concentration, which is optimum for its growth. And a higher salt concentration is not tolerated by non-halophilic Vibrios. Now, in your exams, a lot of MCQs are asked regarding the halophilic as well as non-halophilic Vibrios. So when we say, I have two mnemonics in order for you to learn these halophilic as well as non-halophilic Vibrios, and this gets very easy with it. So for halophilic Vibrios which require salt, I have added a mnemonic. Please add white salt. WHITE has been replaced by VITE. So the examples of these bacteria which require salt for their growth are, firstly we have Vibrio parahemolyticus, Vibrio alginolyticus, Vibrio vulnificus, right? So these are the three bacteria which are three species of Vibrio which are halophilic, which require salt for their growth. And for non-halophilic Vibrio, it is my choice. I do not want salt. I do not want salt, right? So it is my choice. So the examples are going to be Vibrio mimicus and Vibrio cholerae. Vibrio cholerae. It is cholerae and mimicus. So these are all non-halophilic Vibrio examples, mimicus and cholerae, and the halophilic are these three species, right? Moving ahead to new different classification, that is Gardner and Venkatraman classification. So this classification was based on serogrouping, serotyping, biotyping, etc. And majority of you, even I got confused how to remember these serogroups or serotypes, biotypes, etc. So I made a very fun mnemonic, and with this mnemonic, it gets very easy. So most of you might be knowing that Samsung, it is a South Korean company, and these people, they really love rice noodles, right? They love rice noodles, and they have one CEO. They have one CEO. All of these words or letters will be used as, the clue points for your classification. So firstly, we will simply write the classification as GSBS. This is how I learned it, GSBS. So we know what comes first, GSBS. So firstly, we'll have the groups, then we'll be having the serogroup, the biotype, and serotypes, right? So groups are naturally going to be Vibrio cholerae, which is responsible for cholera, right? And non-cholera Vibrios, non-cholera Vibrios. See, this concludes the group. Now comes the serogroup. Since we are talking about rice water stools and the pandemics and the epidemics, so we need to classify which strain, which serogroup or which biotype or which serotype is responsible for these particular pandemics, for very, very important for epidemiological reasons. So we will be classifying Vibrio cholerae, or the one which is responsible for the pandemics, right? So we said that Samsung is a Korean company. So S comes for the serogroup. So serogroup is going to be firstly 1 O1. We said they have one CEO. So O1 is the first serogroup that we will be dealing with. Then we have O139, and lastly we will have non-O1 or O139. So these include more than 2 to around 200, right? So now we said that Samsung company has one CEO. So in bio may we write our designation, right? So CEO comes here. So what is the biotype for that? 1 CEO. So biotype is going to be, there are going to be two biotypes. C for classical and EO for 𝗪𝗏, right? So this concludes the biotypes, that is CEO, right? And we said that Samsung is a Korean company. So Korean and Japanese name, I know, they might sound similar. So we will be using the, Japanese come Korean names, and it will be very easy for us to remember. So the serotypes are going to be 3 for the both of them. So these are Ogawa, Inaba, as well as 𝗘𝗅𝗙𝗏𝗔𝗟, right, for both. Ogawa being the most common serotype, right? And thereafter comes the another serotype, that is Inaba. So now talking about the serogroup. So when we say that serogrouping is done, so for any grouping, as we say, we require certain antigens. For the serological reactions, we require antigen antibody reactions, right? So for serogrouping also we require antigen. So the antigen which we have here is O antigen, which is somatic O antigen, which comes from the cell wall of Vibrio, right? So we are using somatic O antigens for serogrouping. So we said that we have one CEO, the company has one CEO. So we will be describing the importance of these, serogroups when we said we are talking about the pandemics and why are we really classifying this particular bacteria, right? So they have O1, O139 and non-O1 or O139, right? Now, so CEO, O1, it comes first. The CEO is the hero of the company. So he is responsible for all the, he has maximum number of responsibilities, and he's given maximum charges. So O1 has been responsible for all the major reasons, and those reasons are responsible. This serogroup was responsible for all the pandemics worldwide. So it had major role, this serogroup had major role in all the pandemics, and this particular serogroup was responsible for majority of the morbidities regarding this particular bacteria, right? And then we have O139. This was responsible for certain epidemics and outbreaks in coastal areas of India and Bangladesh, but was not responsible for any pandemic. And non-O1 and O139 is not of very huge significance, because it had been responsible for certain sporadic cases only. Certain sporadic outbreaks was not considered to be of much importance, right? But here we need to know the CEO, or one CEO, the first one, was responsible for all the major pandemics. So you need to remember this, right? And then the biotype, we say bio may we write down the designation, that is CEO. So what are the two biotypes that we have already discussed? These are classical and 𝗘𝗏𝗏, right? So all the pandemics were done by O1 serogroup, and now the biotypes. Since we say classical, so classics, they always come first, right? They are going to be hypervirulent. Firstly, the classical biotypes were responsible for first 6 pandemics. They were responsible for first 6 pandemics. They came first, they're classic, and they were also hypervirulent strains. Hypervirulent. They were hypervirulent and they were responsible for first 6 pandemics, right? It was the classical biotype which was responsible for first 6 biotype. These are all questions which get asked in your exams. And then the 𝗘𝗏𝗏. So after first 6 pandemics, then came the picture of another biotype, that is 𝗘𝗏𝗏, which was responsible for the next pandemic, that is the 7th pandemic. And thereafter, this 𝗘𝗏𝗏 biotype, this took over the classic strain for responsible for pandemics. So thereafter, it has been responsible for all the pandemics later on. This has actually replaced the classical biotype after that, and it was then responsible for all the major morbidities associated with Vibrio, right? And then there are 2 important things which are being asked nowadays. Those are 𝗘𝗏𝗏 variants. 𝗘𝗏𝗏 variants, right? So these 𝗘𝗏𝗏 variants are nothing but, these are also known as hybrids of 𝗘𝗏𝗏, and not just 𝗘𝗏𝗏 but classics. So we have 2 variants. One is Matlab variant and another one is Mozambique variant. Mozambique variant. So Matlab variant, this is a mixture, or this is it has properties of both the classical and 𝗘𝗏𝗏 variant. So it's a mix of classical and 𝗘𝗏𝗏, and this is the reason why, because it has so many properties of classical and so many properties of 𝗘𝗏𝗏 biotype. So this could not be typed. So it has been kept under 𝗘𝗏𝗏 variant. So it was not typed because it has the mixture of the properties of classical as well as the 𝗘𝗏𝗏. And Mozambique variant, it has the phenotype and the genotype. Phenotypically it is classical and genotypically it is 𝗘𝗏𝗏. So this also remains to be a hybrid version. So it has both the properties of classical as well as this 𝗘𝗏𝗏. So both of these, because of such properties, they are kept under 𝗘𝗏𝗏 variants. Right? Now moving towards the major differences between these 2 biotypes, we must talk about firstly the major differences. So how do we differentiate these 2 biotypes? And we have done it in our lab, so it gets very easy when you differentiate these 2, and I have used certain buzz letters in order to help you remember the differences between the 2. So firstly, we talk about the cholera toxin gene. So classical, we say classics are always first, they come first. So cholera toxin gene is also going to be CTX1 gene, whereas 𝗘𝗏𝗏, the cholera toxin gene is going to be, for 𝗘𝗏𝗏 it is going to be CTX2. Now beta hemolysis, it would be positive for 𝗘𝗏𝗏 because it also has O. So it is given positive by 𝗘𝗏, but not hemolysis is not seen in the classical biotype, right? And then 𝗃-erythrocyte agglutination. Again, it has E and 𝗘𝗏 has E, so it would be positive for 𝗘𝗏 biotype. Agglutination would be positive for 𝗘𝗏 biotype, whereas it would be negative for classical biotype, right? Then polymyxin B susceptibility. So B and C are the letters which come together. So B comes before C. So polymyxin B susceptibility would be positive, would be given positive by classical. So classical is going to be susceptible for polymyxin B, whereas the 𝗘𝗏 biotype is going to be resistant, right? This you can very easily remember. Polymyxin B, B and C. So it would be susceptible for classical, whereas it is going to be resistant for 𝗘𝗏. Now group 4 phage susceptibility, this you'll have to remember, it is positive for classical, whereas it is going to be negative for 𝗘𝗏 variant. Now when we say 𝗘𝗏-phage 5 susceptibility, it already says 𝗘𝗏 and it has phage 5. So 𝗘𝗏 has 5 letters. So it would be naturally given positive by this 𝗘𝗏, and that will not be the case with classical biotype. And then the VP test, V corresponds with Roman 5, right? 𝗘𝗏 has 5 letters. So it would be given positive by 𝗘𝗏 biotype and negative for classical biotype. So this gives a very clear picture and clear differences for you to remember for your exams, right? So now moving further towards the pathogenesis of Vibrio. Naturally the pathogenesis is going to be by ingestion of contaminated food and water, right? So we know that Vibrio, it is highly acid labile. So in order to get rid of or bypass that acidity of the stomach, it needs to have a higher bacterial load. So Vibrio needs to have a higher bacterial load so that it bypasses the stomach acidity. And in your exams, you're also asked about the bacterial loads of different bacteria, and you need to remember that. So it requires higher bacterial load, right? Higher bacterial load. And what is that bacterial load that is required? It is 10 to power 8 bacilli per ml of stool, right? So how do you remember that? I'll tell you how I remembered it. So cholerae has 8 letters. So naturally this becomes the mnemonic, and this becomes easier for you to remember. It requires 10 to power 8 bacilli per ml to bypass stomach acid barrier. And then it reaches the small intestine, where it is going to release the cholera toxin, which you must remember, it is a type of enterotoxin, which would be responsible for all the pathogenesis of these rice water stools. So this enterotoxin, it is similar to the 𝗕𝗵𝗏𝗏𝗏 of Escherichia coli, right? You must remember this. Now we said the bacteria, it reaches the small intestine, and it is in the small intestine where it is causing the release of the enterotoxin. But how does that do it? So firstly, this Vibrio is going to penetrate the mucus layer of the small intestine, right? And it is at the end, after penetrating the mucus layer, what is it going to do? it is going to reach near epithelial area, epithelial lining, or small intestinal epithelium area, right? And in doing so, it is going to release certain proteolytic enzymes, certain mucinases, so that it cuts down that mucin in order to get attached. So what next it is going to do? It is going to release mucinases, proteolytic enzymes, right? So it clears the mucus, fibronectin, etc., clears the mucin, and then it is going to get attached to the epithelial lining, to the intestinal epithelium. And how is that attachment happening? Again, a very important question that happens with the help of a pilus, that is toxin co-regulated pilus, This is the pilus with which it gets attached, and after attachment it is going to release the cholera toxin, right? So now, talking about the cholera toxin mechanism. So lot of bacteria have different mechanisms for their pathogenesis, and also they have different receptors what attaches where. So in order to ease it for you, so I have another fun mnemonic. So Vibrio has all the Koreans and Chinese and Japanese mnemonics, so, and that too related to food, so it gets easier that way. So for cholera toxin mechanism, I have a mnemonic that is big Asian chopsticks. You'll know how we use it now. Firstly, we must know that enterotoxin has two fragments. It has two fragments, fragment A and fragment B. Fragment A and fragment B. Now A is the active fragment and B is the binding fragment, right? So firstly, when we said we take the first letter B, so this binding fragment, B fragment, gets attached to GM1 ganglioside receptors. On the intestinal epithelial area, right? So B and G have been used. Now, after attaching this A fragment, this active fragment, it is internalized. Fragment A is internalized. Now, this after internalization of this A fragment, what causes? ADP glycosylation. Now, we are using the letter A. ADP ribosylation of G protein occurs. Now what happens next after ADP ribosylation? Again, it increases the activity of, it upregulates regulates the activity of upregulates the activity of another A, that is adenylate cyclase. Now, due to the upregulation of adenylate cyclase, what happens? There is going to be an increase in, now we have used two A's, so there is going to be an increase in cyclic AMP. There is going to be an increase in cyclic AMP, and with increase in cyclic AMP, two things happen. Two things are going to happen. Firstly, the absorptive sodium channel is going to get inhibited. Absorptive sodium channel is inhibited, right? And the secretory chloride channel, it is going to get activated. So absorptive channel is blocked and secretory channel is activated. So there is going to be, what happens thereafter? There is going to be increase in sodium chloride concentration. There is going to be an increase in sodium chloride concentration. Now, increased NaCl, it is naturally going to, attract lot of water. So, passively, lot of water comes into the lumen, and later becomes very, very voluminous. Because it needs to maintain the osmolality. Voluminous. So this is the reason why we have rice water stools. So there is going to be excessive water, increased rice water stools. This is the whole pathogenesis. This is the cholera toxin mechanism, which explains us why is there so much water in this type of diarrhea or Vibrio cholerae diarrhea, right? So we can very well see that with the help of this picture. So this is the Vibrio cholerae. We said it has two fragments. This is fragment A and this is fragment B. So B is the binding fragment. This is cholera toxin, right? B attaches to GM1 ganglioside, right? So this is GM1 ganglioside. So B is going to attach here, and then after attachment, A subunit is going to get internalized. And after, when it gets internalized, what is it going to do? It causes ADP ribosylation, right? And ADP ribosylation, it upregulates the activity of adenylate cyclase. And in doing so, what it causes? It leads to increased cyclic AMP. And increased cyclic AMP ultimately leads to increase in chloride, increase in sodium, and increase in water. So increased sodium chloride and high voluminous and increased amount of water in, right? So this is the whole mechanism of cholera toxin. Now this is the rice water stool. This is the most probable picture that you will be getting in your exams, rice water stool with appropriate history, right? Now moving towards you should also know what are the genes which are responsible for the virulence and for the mechanism, the toxin release and everything. So these are the three genes you must remember for your exams. One is cholera toxin gene. We have already discussed that. Cholera toxin gene, which would be responsible for virulence. Then we have the zona occludens toxin. It is going to disrupt the tight mucosal junctions, which again would be responsible for increased volume of water and increased fluid, passage. And then we have another toxin, that is accessory colonizing factor. Accessory colonizing factor, which is responsible for or which helps in adhesion as well as colonization, right? Now moving on to the clinical manifestation. Firstly, when we said about Vibrio cholerae diarrhea, it is an acute state. It is responsible for acute diarrheal disease. So the incubation period is naturally going to be 24 to 48 hours. It is naturally going to be 24 to 48 hours. And the kind of diarrhea which we said, it is going to be watery diarrhea. So it will be painless diarrhea, high volume, and it is passive in nature. Passively comes in the lumen. So painless, high voluminous, passive diarrhea is seen as watery diarrhea in Vibrio cholerae diarrhea. And the typical rice water stool, how we describe rice water, and this picture shows that as if the rice has been washed away and the remaining water, it looks exactly like this, and the stool also looks like this. So it is going to be cloudy stools, non-bilious, and mucus flakes would be present. And they are not associated with any fecal matter. It is just the water that comes passively because of the sodium chloride, so it is not associated with fecal matter, and no RBCs. And for watery diarrhea, no RBCs or pus cells would be visible under microscope, right? So they are not associated with any fecal matter, and also they have this type of stool has offensive, offensive is the wrong word, it has a type of fishy odor. And why is that fishy odor? Because of the metabolic byproducts, because of large amount of lot of bacteria are responsible for causing this type of diarrhea. Higher bacterial load is required, so the byproducts are naturally going to be high. So that is the reason why we have fishy odor in rice water stools. So also when we make a gram stain of the mucus flakes, so what do we observe? A typical morphology or typical pattern is observed, and it is also a question for your exams. So see these bacteria, they seem to be moving in parallel rows and in one stream or in one line. See, majority of the bacteria are seen. So this is the typical arrangement seen, that is fish in stream appearance is seen in Vibrio cholerae diarrhea or rice water stool, and this picture depicts the the fishes moving in one single line. So this is the picnomic for fish in stream appearance, a very potential question for your exams. So this was about the selective very high yield points from this particular bacteria. So this was all about Vibrio. Thank you.